This method can help answer key questions in microbial ecology and biotechnology, for example, which ecological concepts drive any given ecosystem. The main advantage of this technique is that it can be used to closely follow rapid microbiome dynamics with a short interval sampling regime while remaining highly cost-effective.
This method can be used to get information on biotechnological and natural microbial communities, and also on animal and human microbiomes for nutritional and health states.
Demonstrating the procedure will be Florian Schattenberg, technician and cytometer operator in our laboratory.
To dry a biogas community sample, use a modified 1 ml pipette tip to transfer 200 microliters of the viscous digestate to a 2 ml tube containing 1.7 milliliters of PBS. After thorough mixing, place the tubes in an ultrasonic bath for 1 minute to disband any large cell aggregates and detach cells sticking to plant cell residues. After the sonication, mix the sample thoroughly and filter the cells through a 50 micrometer pore mesh strainer into a 2 ml plastic tube. Divide the filtrate into 4 400 microliter aliquots and centrifuge the aliquots 2 times, discarding the supernatant completely both times to mechanically dewater the microbe sample as thoroughly as possible. Then dry the sample in a heated vacuum centrifuge to create a stable pellet, and store the pellet at 4 degrees Celsius protected from light. For stabilization,